Well, I first want to thank all of you for being here and coming from different parts of the world and contributing to what has been a really interesting meeting. And I'm very pleased to have the opportunity to present some of our work today. And I'll basically be following up a little bit on what Garrett was talking about. So the topic is kind of general active dynamics and cellular mechanosensing. And we've been working on trying to understand how cells sense and respond to their physical environment in many different contexts. And that's sort of been the theme of what we've been doing in our lab. So just to start you off, I wanted to show you a movie where this is a larger scale example of cell sheets kind of moving. These are cells from neural crest, from the neural crest in chick embryo. And this is an in vitro experiment where you can see that the cells are moving out of this explant and they interact with each other, change their shape, and you can see there's a lot of potential pushing and pulling going on. What you can't see, but what is inherent is that they're also interacting with their environment. So it has been found that cells can sense the mechanical properties of their environment, and they exert forces, and they can also respond to forces. So typically a lot of work has been done in understanding how cells respond to chemical features of the environment, growth factors, chemotactic features, etc. But over the last couple of decades, it's become increasingly clear that cells can sense physical cues. So, for example, in this really remarkable paper in Dennis Disher's lab, they showed that stem cells could differentiate based on the physical property, in this case the matrix stiffness on which the cells were grown. So the softer substrates they would differentiate to brain-like cells, on stiffer substrates they would differentiate to bone-like cells. So not only stiffness, but for example surface topography can have a remarkable effect on the shape of the cells, as you can see on a flat surface versus a 3D collagen matrix. And this has also become increasingly studied in the last several years. And so these physical cues can determine the biochemical and genetic responses of cells. And an underlying question is, how does the cell do this? So one way that the cells have to respond to their surroundings is to exert forces. So they can push and pull and tug on the surfaces, reorganize their shape in response to surfaces, and thereby sort of change their behavior. So just sort of very quickly what Garrett talked about in more detail, you can have many components in the cell that contribute to cellular forces. Molecular motors, so in this case, for example, myosin motors walking on active filaments and pulling filaments together to contract them. Polymerization of actin, of course, actin assembly and disassembly can lead to protrusive forces. And what are also important is the structure of the actin network. So there are a diverse array of actin cross-linkers that act as glues. They bind actin filaments together, and it can lead to diverse structures, for example, bundles or meshworks. And this network architecture can determine what kind of forces, for example, molecular motors exert on that particular architecture, and also can determine how polymerization affects cellular force generation. So all of these forces together must then balance and then create a situation where people have studied the classic examples of cells crawling on the surface, on an adhesive surface. So some of the work that we've done, just wanted to point out that, as I mentioned, architecture is important, and in many kinds of cells, typically adherent cells, epithelial cells, fibroblast cells, the actin filaments organize into higher order structures, such as stress fibers. In this case, there's sort of cartoon examples of 3 different kinds of stress fibers: ventral, transverse, and dorsal stress fibers that span the cell. They originate at its sites of adhesion called focal adhesions, which are also sites of force transmission to the extracellular matrix. by virtue of adhesion proteins such as integrins. And so these contractile bundle stress fibers are also very important in force generation and force balance inside the cell, and I'll talk about this in more detail later. There's sort of a few different levels at which these forces can enable cells to sense and respond to forces, to the physical environment. So at the level of single proteins, you can have force-dependent conformational changes of proteins. So for example, protein receptors, and we've studied this in immune cells. Or you can have force-dependent aggregation of proteins, for example, in the formation of these adhesion structures, focal adhesion structures, or again, signaling clusters in immune cells, or you can have forces that are acting over the entire range of the cell. So actually, this is taking place at multiple scales, and that's one of the challenges to try and understand how you go from the level of sort of single proteins to the whole cell, whereas the whole cell responds to the mechanical environment. So one of the questions that we're interested in answering is how actin networks are assembled and organized in cells, and how the properties of actin networks themselves modulate force generation and mechanosensing. So in the last couple of years that I've been here, I've talked mostly about our work on immune cells, and just sort of point out that immune cells form junctions with other cells. So for example, T cells form junctions with antigen-presenting cells. They have to recognize the pathogenic peptides and antigen-presenting cells is spread out on these cells, and in some cases they go on to further action. For example, cytotoxic T cells will end up killing the other target cell. So some of our work has focused on looking at the dynamics of the cytoskeleton. Here you can see it's a very different sort of amorphously organized actin network, which shows dynamic actin waves rather than these more organized actin bundles that you see in adherent cells. However, these actin networks can also exert forces, as we measured in the last few years, and we are trying to get a better understanding of how these forces are coupled to the signaling function of these T cells as they become activated. And we also showed that these cells and the forces they exert are sensitive to substrate stiffness. We also studied B cells, which are another kind of immune cells, and we found that B cells are sensitive to ligand mobility, and we are trying to understand their response to surface topography. And as my student Ivan talked about, we are interested in looking at the single molecule analysis of the B cell receptors. So today I'll focus on a different type of cells that we're working on, and where we're looking at cells of the cancer microenvironment that are derived from pancreatic cancer. So as several of the talks have pointed out, and this is becoming increasingly known lately, that the cell's ability to sense and respond to mechanical cues is important in many diseases, including cancer, the spread of cancer metastasis, and the cancer cell's ability to sense the external environment, as well as other types of diseases like cardiac hypertrophy or atherosclerosis. And alterations in these oncogenic activity can lead to cytoskeletal reorganization and changes in cell and tumor stiffness. So we're focusing on these cells called tumor-associated fibroblasts, which were derived from a pancreatic tumor, and asking the specific question of how actin cross-linkers modulate cellular actin architecture, and how is this important for the mechanosensing ability of cells. For example, this cross-link expression in cells regulate adhesions, actin structures, and forces. And I'll also show you some recent work to understand how the dynamics of actin and actin cross-linkers are modulated by the nanotopography of surfaces. So basically focusing on sort of two different types of physical cues that encountered by cells in vivo, the stiffness of the environment, and the topography of the environment. So we focus on this particular actin cross-linker pallidin, which is very important in cells. It's essential for embryonic development in maintaining cell shape and motility. It has been found to be upregulated in pancreatic tumor cells. And it's also an integral component of these actin bundles I mentioned, the focal adhesions and stress fibers. So it co-localizes with another well-known actin cross-linker, alpha-actinin. Pallidin and alpha-actinin are mutually interacting, and they also bind to actin filaments. So here you can see pallidin, alpha-actinin, and sort of a co-localization of them. In some earlier work, in vitro work, we showed that pallidin and alpha-actinin synergistically modulate in vitro actin network viscoelasticity. And so we wanted to look at its effect in cells. This is just a little bit more detail about the structure of pallidin. So we constructed cell lines using that label the pallidin with GFP, and if we allow these cells to just spread out on a fibronectin encoded surface. Okay. So you see initially that the pallidin is organized fairly amorphously, and then as the cell spreads out, it after about 10 to 15 minutes of spreading, it organizes into these stress fibers, and it's found sort of present as sort of punctate structures on these stress fibers. Looking kind of a little more closer at the earlier stages, you can see that pallidin, which is in green, also localizes to focal adhesions and co-localizes to some extent with paxillin, which is a classic focal adhesion protein. But the pallidin and paxillin actually show distinct kinetics as they organize in these focal adhesions, and initially you have pallidin at the leading edge of the cell, whereas paxillin is in the interior. They become coincident, and after several minutes, the paxillin is at the leading edge and the pallidin goes interior to the cell and starts getting involved in formation of the stress fibers. So with the help of our collaborators at University of North Carolina, Chapel Hill in Carol Oates' lab, we constructed these cells that were knocked down in pallidin so we could modulate the expression level of pallidin. And we found that knocking down pallidin has an effect on the focal adhesion development. You get shorter focal adhesions in pallidin knockdown cells, and also smaller maturation time of focal adhesions in these knockdown cells. So pallidin, we found pallidin to be important in focal adhesion formation. As I mentioned, pallidin associates with stress fibers, and as you can see from this intensity profile here, it forms these distinct bands across the focal adhesions. So this is architecture that is been observed many times before. And here we label together pallidin and myosin, and as you can see, myosin also has this sort of punctate band-like structure. If you look at the intensity profile, we see that the pallidin and myosin alternate in these bands. So this is sort of the sarcomeric structure that people have observed, but it's not very clear how this emerges. But both of these pallidin and myosin bands have a similar spacing, about a micron apart. The knockdown of pallidin cells also impairs the formation of what are called radial stress fibers. As you can see, these are radially emanating between the cell periphery and closer towards the cell center. The knockdown cells don't or have an impaired ability to form these stress fibers. So since pallidin is involved in focal adhesions and stress fibers, which are sort of these force-generating structures in cells and force-transmitting structures, we wanted to test the role of pallidin in the mechanosensing of elastic substrates. So we did that by using traction force microscopy. We allowed the cells to spread on a soft gel with beads as fiduciary markers. And as the cell spreads out, exerts forces, the beads move, and then from the displacement map of these beads, if you know the elasticity of the matrix, you can get the traction forces that are exerted. And the advantage of this technique is we can change the stiffness of the substrate to then see how the cell responds to changing stiffness. So this is a picture of the EGF-repellated cell and the traction forces, the stresses it exerts. And you can see the forces are centripetal, as has been observed for many different cell types. What we found that these cells, and here we're just showing the EGF-repellated and wild-type cells, they behave very similar to each other. They do exhibit a sensitivity to substrate stiffness because the forces increase as the stiffness of the surface increase from a few kilopascal to about tens of kilopascals. And you can see the organization of actin is very different, with a more amorphous organization on the softer gels and a much more bundled structure. As I showed you, the previous pictures that I showed you were on glass. So for the stiffer gels, they become closer and closer to resembling what they do on glass. So what happens when we knock down palladium? What we found remarkably, and a bit to our surprise, was the knockdown cells, which is shown here in the lighter gray, exerted much greater traction forces than the wild-type cells or the EGF-repellated cells. So decreasing the concentration or decreasing the expression level of these active cross-linkers increased the forces and also changed the manner in which the cells responded to the surface stiffness, because at higher stiffnesses, again, there was no difference between the forces. So clearly the expression level of this protein makes a difference. So we wanted to probe this a little bit more dynamically. So we first used blebistatin, which is an inhibitor of myosin 2A activity, and most of the forces in these cell types are derived from myosin. So when we add blebistatin, you see almost a complete disappearance of the forces, and then we allow the cells to recover by washing out the myosin, and you can see the recovery of forces. So what we find here is that, again, the knockdown cells, which are shown here in green, show much the knockdown cells are shown in red, show a faster recovery. So this shows a stress recovery. Sorry, I seem to have lost the unit somewhere at the bottom. But this is in minutes. So the cells recover on the order of 15 to 20 minutes. So these knockdown cells show faster recovery to their original forces compared to the wild type or EGF-repellated cells. So we find these distinct dynamics of force in cells that are knocked down in the active cross-linker palladium. Does this imply distinct active myosin dynamics in normal versus the knockdown cells? And that's not something that we have. So the summary for this part is that we found that the active cross-linker palladium does have a strong effect on force generation and mechanosensing ability of these tumor-associated fibroblasts. And we are thinking, could this potentially imply a cooperative action of active cross-linkers in myosin being important for cellular force generation? So if you have lower amount of cross-linkers, in what way does this affect myosin's ability to generate contractile forces? So for the last few minutes, I'll talk a little bit about some of our very recent work on trying to understand the response of these cells to a different physical cue, surface nanotopography. So why are we interested in surface nanotopography? Because cells encounter extracellular matrix in vivo, and these extracellular matrix surfaces are almost never completely flat. And again, there's been an increased interest in understanding how cells behave differently in different kinds of topographic surfaces. So, for example, collagen fibers can be several hundred nanometers wide. They can be arranged in sort of a random meshwork, or they can be arranged in more parallel ordered fibrils depending on the kind of environment the cells are in. So, There's been some amount of previous studies on surface nanotopography, looking at collective cell behavior, single cell behavior related to differentiation, cell shape, migration, proliferation, and people have used these kind of grooved surfaces or nanopore surfaces. So we've been working in collaboration with Dr. John Frotus's lab at UMD, and they've been patterning these nanotopographic surfaces with very high precision. So these are a set of parallel ridges or grooves, which are separated by a certain space that we call the pitch or the spacing between them, which range from about 0.8 microns to 10 microns. And these grooves are about a few hundred nanometers in height and 250 nanometers in width. So we allow these cells, the tumor-associated fibroblast cells, to spread out in these grooved surfaces, and here I'll just show you a movie of the cells spreading dynamically. So as you can see, as the cell spreads out, sort of seems to preferentially elongate in the direction of the ridges and appears to be more aligned. So we quantify this. We do see that cell alignment, in this case the orientation of the cell or the elongation or the aspect ratio of the cell, is significantly greater for grooved surfaces. These show the range of surfaces that we use. And again, this parameter, for example, seems to peak around 3 micron, a surface with 3 micron pitch or spacing. So we also found that stress fibers that I showed you, so this shows a flat surface, a 3 micron surface, and a 5 micron surface. The stress fibers align with the ridges, and we quantify this with a stress fiber order parameter, which shows that again, there's most significant alignment on this 3 micron surface, as we can track the stress fibers as shown here. We also found that the focal adhesions align with these ridges, but also very interestingly, the focal adhesions appear to mature on the top of the ridges. So you have longer and more numerous focal adhesions on the top of the ridges compared to the spacing between the ridges. And this shows a comparison of the length of focal adhesions on the flat surface when compared to the bottom of the ridges and the top of the ridges on a 3 micron surface. So these are some of the static parameters that appear to be dependent on surface topography. But what we noticed from some of the movies that I showed you earlier, and I'll just play this again, I'm able to, and now it seems to be lost. Okay. So this is the movie I showed you earlier. The cells form these stress fibers, and there's this intrinsic dynamics of the stress fibers in the cells. There's been a lot of interest again also on intracellular dynamics in cells and what these are controlled by, and we wanted to test how this is affected by the surface topography. So this shows a similar type of cell, EGF repellated, on a 3 micron pitch surface. You can see the cell is very active. The edges are moving, the whole cell is sort of going through a movement. And if you look at the interior of the cell, you see the stress fibers are showing these very interesting dynamics. And what we can do is use these bands of palladium as fiduciary markers. They form these punctate dots. We can use them to track the movement of the stress fibers and from them obtain these sort of tracks of the move, that is a signature of the stress fiber movement. So these palladium dots provide us with a unique way to track the actin dynamics. We found that the palladium motion is directional, becomes directional on these patterned surfaces. So this shows an example track on a flat surface and the distribution of the angle. So this is the angle between the displacement or any displacement of the track compared to the direction of the ridge. This is the direction of the ridge. On a flat surface, you see a uniform distribution. this is a typical 3 micron surface, you see that it becomes much more directional, and that is represented here. The width of this distribution is narrowest again for this 3 micron pitch. We also found that the velocities, the instantaneous velocities, were higher for all pattern surfaces. This is shown the control of the flat surface, shows the histogram, the black histogram, and the green histogram is for the distribution of velocities on the 3 micron surface, and you see the emergence of this longer tail, and the same thing is shown in this cumulative probability density. Again, the 3 micron surfaces show the higher velocities. And this shows the dependence of the average instantaneous velocity with the direction of the ridge. And again, you see this strong peak with a 3 micron surface, but a much flatter distribution with a flat surface. And then we analyzed the particle tracks that I showed you earlier. And what we found is that all of these tracks show a superdiffusive motion. So we fit it to this type of function to get an effective diffusion coefficient and look at the exponent of the movement. We found that the Paolo exponent was between 1.65 and 1.8 for all the patterns, for the flat as well as the ridged patterns. But the ridged pattern showed greater MSDs and a higher effective diffusion coefficient. So finally, in conclusion, we found that the cells respond to surface topography in a somewhat biphasic manner, showing the greatest amount of response for these 3 micron pitch surfaces. We don't know the significance of this length scale yet, and that's one of the things we're interested in figuring out. And what are the underlying mechanisms of this topography sensing? So a lot has been done on cell shape and differentiation and more static aspects, but how the dynamics of the cell itself modulated by topography? Could this be related to curvature sensing proteins? Or what are the underlying signaling pathways that are involved? We also think that tracking these kind of palladium bands could provide a somewhat unique and non-intrusive method to measure these intracellular forces that might drive the dynamics of structures inside the cells. And we are working on probing the origin of these intracellular dynamics. We already know that these are related to actin assembly, but that they're not ARP2/3 mediated. They are definitely myosin dependent. And we are also working on studying the effect of the substrate stiffness on these intracellular dynamics. And finally, just to come back to what I showed earlier, so it's becoming clear that actin cross-linkers and actin organization is important in cellular mechanosensing. As I pointed out earlier, for example, you see these sort of very interesting actin organization inside cells, and I'm just going to present a very preliminary movie from Garrick's lab, because Garrick's group has been working on modeling these kind of structures that include all the components of actin, myosin, as well as cross-linkers. And this is a very preliminary movie, but it shows about a 1 micron length of actin filaments that are being acted upon by myosin in red and actin cross-linker, I think alpha actin in yellow. And it shows that you can put these components into the modeling, that then we can start to predict, hopefully, and sort of facilitate new experiments and motivate new experiments to try and understand what the underlying mechanisms are. So with that, just going to skip over this, some of the other stuff that we're interested in, and acknowledge my lab for all the work they've been doing. The work that I showed was done by Mike Azatov, a student who recently graduated. And these are all my students working on the different projects in my lab. And this work was done in collaboration with Dr. Carol Odie, and I'd like to thank the funding sources and also calls for this meeting. Thank you for your attention.
At most one question.
So the palladium-associated focal adhesion dots on the ridges, do you always see a dynamic dynamic structure? I mean, they are moving, or do you also see a stationary dots, and does that change with respect to the pitch of the ridge?
So the palladium dots that we see moving, they're on stress fibers. But palladium also localizes to focal adhesions, and those are more static. So the focal adhesions are not necessarily moving on the ridges. It's the dots, and that's reflective of the stress fiber.
Thanks, Dr. And the rest of the panel.